Hello, I'm Tim Wall, Senior Reporter for Pet Food Industry. Thank you for joining us for this edition of Ask the Pet Food Pro, Pet Food Processing Controls to Inactivate Avian Influenza Virus. Ask the Pet Food Pro offers industry professionals the opportunity to pose questions directly to experts. Ask the Pet Food Pro is brought to you by Pet Food Forum and Pet Food Industry. Drawing on our journalists and editors' combined decades of experience covering the dog, cat, and other pet food markets, you can find our reporting and analysis at petfoodindustry.com. Don't forget to subscribe to our daily newsletter and monthly magazine. Today's discussion focuses on how to kill bird flu in pet food ingredients. Extrutech sponsors this edition of Ask the Pet Food Pro. Our expert guest is Dr. Mary Grace Denow, Research Associate Professor in the Department of Food Science and Technology at the University of Nebraska, Lincoln. Welcome, Grace, and thank you for joining us today.
Thanks, Tim. Thanks for inviting me. I think this is a very timely topic, but I do hope, just like everybody else on the call today, they realize I'm also just learning about this topic as well, but I'm always happy to share what I've learned, and hopefully that also can help someone out.
Thank you. So before we start the discussion, I have a few announcements. Audience members, you have all been muted to ensure that everyone can hear and focus on the discussion. I've included many of the questions that you all sent in beforehand into our script, but if you have questions during the discussion, please submit those or any comments in the chat box. Please note that this chat is being recorded, so you can access the recording starting in about a week at petfoodforumevents.com. Click on the Ask the Pet Food Pro tab up at the top. So, since last year, it feels like I haven't gone more than a couple of weeks without reporting on another correlation between pet food and bird flu infecting a domestic cat. Before we start talking about how to address this problem, Grace, as a scientist, could you give us a 101 lecture on how avian influenza can pass from raw pet food to a pet cat?
Okay, I will try. So typically H5N1, like other influenza A viruses, we would think of it as a respiratory illness. And so we don't normally think that you can get the flu when eating food that is tainted or contaminated with a flu virus. However, it's been documented in the literature that there have been some large cats or outdoor cats that have gotten sick and maybe eventually die as well once they've kind of gotten into contact with infected wild birds or waterfowl. Certainly, there's an assumption that if they were also fed with virus-infected poultry meat, that they would also succumb to illness. So it wasn't until like around 2010 when somebody actually did a study to demonstrate or to, I guess, to test whether or not is the GI tract actually a portal for entry for the virus for domesticated cats, meaning, again, if they ate something that was tainted or contaminated with an influenza virus, can it actually pass through the kind of like the digestive tract, somehow move in to, let's say, their circulatory system and start to infect the rest of the body? So if anybody's interested in seeing that research or that particular paper, it came out of 2010, and that was the very first time it's been demonstrated. And then since then, people started to become more aware that this could be a route for infection. So people are starting to test more for it. It seems to be cats of all sizes are particularly sensitive to this, but we don't necessarily see that as a pathway. So far, there's not been a whole lot of data when it comes to other mammals, like, let's say, our companion animal dogs, for us humans, for ferrets, and the like. So it is possible and such. So.
Gotcha. So the science is in that it is possible, and we're seeing it in the real world.
And we're testing for it more. Yeah.
And it's definitely on the radar. So we've especially seen this in relation, obviously, to poultry-based pet foods, it being a bird flu. So which types of poultry-based pet food ingredients are most at risk of HPAI contamination? For example, are eggs a concern? That was a question that came in earlier.
That came in. Okay, yeah. No, again, that's a very interesting kind of aspect also when I was doing my research on this particular topic. So, when we talk about avian influenza viruses, one of the first things that I learned was you can have H5N1, but you can also have two subtypes of it. You can have the low pathogenic strain, and then you can also have the highly pathogenic strain. Both strains, and especially the low pathogenic strains, we see it more accumulate, obviously, in the respiratory tract of the animal. So lungs, if you will, would be affected, the diaphragm, if it was a mammal. But then it also tends to accumulate in the digestive tract because that's another route for infection. When we're dealing, however, with highly pathogenic avian influenza virus, the only thing that's different between LPAI and HPAI is how fast the HPAI strain can actually replicate. So then the whole idea is that virus just grows in numbers so quickly that it can overwhelm the host. So then in this particular case, it's some kind of a poultry animal, right? That will be its ideal host because it's bird flu. Then this virus spreads so quickly, it now no longer is just contained within the respiratory and the digestive tract. We can actually see it in the bone marrow. We can see it in some of the additional other organs, for example, like livers, which are a very popular item or ingredient in pet foods. And certainly you can also start seeing it in the muscle tissue if it's gotten that far along in the illness. So I think overall, because we're dealing with a highly pathogenic strain right now in the US, it's hard to say that nothing within like a part of a poultry animal or its byproduct will be free of the virus. I think right now, any part of the animal can be assumed to have the risk for containing the virus. When it comes to eggs, because that was a very specific question, we do have validated procedures to pasteurize eggs in shell. I don't know if it's good news or bad news, but some of the papers that I've read, typically for egg laying hens, if we're dealing with HPAI, the highly pathogenic strain, the disease overcomes the animal before it can actually lay an egg that has the virus in it. So I think that in addition to ensuring that you use a thermally pasteurized egg, should hopefully reduce the risk for the virus being active in if it were in the egg.
Gotcha. And so speaking about some of the other tissues that might carry the virus, can
Mhm.
those ingredients be effectively tested for HPAI before entering the pet food ingredient stream?
Yeah, so testing right now is really, I think, one of the biggest challenges when it comes to this virus. It's not a common foodborne virus like we would think of, let's say, noroviruses or Vibrio. So a lot of our commercial food testing laboratories do not necessarily have the tools to deal or to monitor and test for this virus in large numbers, especially. I know a lot of the PCR test kits that are available and are used by veterinary diagnostic centers that have been involved in some of the testing of pet foods here and there since January. They know that those PCR test kits were designed for animal testing. So I guess, yeah, I think if you were, what do you call this? If you were using singular animal proteins, so for example, if you're making treats, then it is possible, technically, for you to get a PCR test on that particular ingredient and then double check and see if it has the virus in it. However, testing of ingredients and pet foods itself, it really depends on whether or not this whoever is in your state, the veterinary diagnostic center for your state, is allowed to do so. So I think that's the trick because we have a very active outbreak right now. So most testing is really prioritized to detect infected flocks. And so human food, pet food tend to be a little bit lower. Because we're seeing it in cattle, we've now also prioritized testing it in milk because the dairy cows are shedding large amounts of this virus in the milk, which is also why I want to highlight that it's really problematic to drink unpasteurized milk at this time, just because they really are going to be in large numbers if the milk is coming from an infected cow.
Actually, I think there even was a case of that in California of a cat.
Yeah. Yes.
So speaking of this current outbreak, is there an explanation as to why in the past few months we've really seen this increase in cases of HPAI from cat food?
Yeah, I don't know exactly the reason, and I think it's also a question that a lot of our people in our audience probably also have. I think of this maybe as being twofold. Number one, we've been in this outbreak now of avian influenza for quite some time. We see, when you're dealing with viruses, my colleagues in animal science, veterinary science, and also virologists have told me, yeah, viruses just replicate like very, very quickly. They mutate, also share DNA with other um, avian influenza viruses. So, because it's an ever evolving issue, you can kind of maybe wrap your brain around this, kind of like what we experienced during COVID. For some time, we kept seeing different variants popping up here and there. So, that's also happening with this particular avian influenza virus, even though they're all H5N1, there could be some variants. Certainly, we're also looking into the literature, and I found some papers that say it is possible for some birds to be asymptomatic of the virus if they got infected within the first few hours of contracting the virus. So even though we're testing flocks, we're not able to test every single bird for the virus. So it could be possible, I suppose, if we're seeing some birds starting to show signs of ace that are there asymptomatic in the first few hours after infection. But then I think also the heightened monitoring of the for the virus and the fact that everybody is on alert, that cats are very sensitive to this. So the more we suspect that a cat has been infected, then yeah, it's our due diligence to test for it.
Gotcha. Now, speaking of that, and in the face of all those risks, let's discuss a little bit more about what pet food makers can do. So first off, what about conventional pet food extrusion? How does that inactivate the bird flu virus, and what are some of the critical points in that process? Just for example, I know in South Korea, I think last year, maybe 2 years ago, there was a situation where a malfunctioning system allowed some of the virus to pass through, even in an extrusion process. So
Yeah.
how about that? How about extrusion?
So I think overall, I think this is where I can, maybe provide some good news. It's well documented in the literature. Several studies have demonstrated that so long as you have a thermal process, and then it's validated for your bacterial pathogens like salmonella, right? And so if we look at some of our FSMA guidance documents for, what pressure, temperature, and time combinations during extrusion cooking, if you're using those to create your extruded pet food product, viruses tend to be wimpier than bacterial pathogens. So if you're able to control and get a 5 log reduction in your salmonella, you're also effectively inactivating the virus. So, and I think that goes almost across the board for all thermal processes where there's been a lot of validation data that says, what temperature, time, and pressure combinations get you that kill in salmonella, then you could be rest assured also that would kill the virus. I think the one thermal process that I would hesitate saying that to would be, let's say, sous vide cooking, where more and more people, because of their desire to have something minimally processed, we don't have as much data, I think, when we're looking at, let's say, lower temperatures and longer hold times at those temperatures, whether or not that would be sufficient to kill the virus. So,
Gotcha. So getting a little bit more kind of into the weeds on that one,
mhm.
is there documentation or do we know what processing parameters, time, temperature, pressure, are necessary to inactivate the avian influenza virus in raw poultry products?
Yeah, so for this one, we actually have a really good reference, I think. I would encourage everyone to look at the World Organization of Animal Health, or WOA, I think is how people pronounce their acronym. They actually have a table on temperature time combinations if you're using a thermal process to inactivate HPAI in meat products. So, for example, if you want to process one of the combinations that they specified on the table was if you wanted to process at 60 degrees C, so that would be 140 degrees Fahrenheit for those of us in the US, you will need about 507 seconds or 8.5 minutes of holding it at that temperature in order to inactivate the avian influenza virus. Now, if you are looking for a short holding time, you will have to go all the way up to about 74 degrees C or 165 F. For that one, when you reach that temperature, you need to hold your product for half a second in order to inactivate the avian influenza virus. What's really great about this particular number, 165 F for half a second, it tracks very well with our recommendations for properly and fully cooking um, poultry meat to inactivate salmonella. So again, this is kind of like probably when you look at the CDC webpage or even USDA FSIS webpage, they are always recommending folks to properly and fully cook your poultry items to safety, meaning 165 F for 1 second in order to kill salmonella. And within the first half second, you would also be inactivating the avian influenza virus. So there's other things in WOA that I think are kind of interesting in discussion about this virus. The Food and Agriculture Organization, or FAO, uses these numbers to also make recommendations globally in terms of ensuring the safety of cooked poultry products. So.
Gotcha. And just kind of a little tangent off of that, when we're talking about wet or canned pet food,
Mhm.
what role does pasteurization play in controlling HPAI?
Yeah, so again, this is another time and true tested process. Makes my heart as a processing engineer great. So if you have a validated process, and a lot of folks I know in the pet food world have taken or gotten certified in better process control school, everything that you've learned throughout that training that showed you what pressure, temperature, and time combinations help you inactivate Clostridium botulinum and its spores. That's actually a quote unquote fairly aggressive treatment during retort cooking or canning that will also inactivate salmonella, and in this particular case, also AIV. So it's almost, I want to say, do what you're doing now, because so long as, I assume you have a validated process to kill those pathogens, you're also effectively killing the virus.
Gotcha. So it sounds like with kibbles, got some protect or some processes there, and with wet foods as well. But
Mm-hmm.
a lot of the cases, or in fact, all of the cases of cats being infected with HPAI from poultry ingredients have involved raw pet foods. So let's
Yeah.
kind of focus in on that a little bit. Can high pressure processing inactivate avian influenza in cat food diets?
Yeah, for most people who know me, this kind of enters my world and such. So high pressure processing is interesting. Like you mentioned, you're not able to achieve high temperatures in high pressure processing typically. I think at most you might be at room temperature, maybe all the way up to 30 degrees C. But those temperatures are not going to be enough to kill the virus alone. So we have to rely on the combination of high pressure and holding time in order to inactivate the pressure. So one of the things that we kind of think of, or what I've told people when it comes to HPP, let's think about its mode of inactivation. Like, how does HPP actually really work? So it works at killing pathogens, because that's what we've used it mostly, is to control for Salmonella, Listeria monocytogenes, and E. coli 0157H7 and other STEC in pet foods. So, how do those bacteria die off? Usually, when you pressurize, and in the US, some of the more common processing conditions are a combination of 87,000 psi for 3 minutes. That whole process, what it does is it starts to unfold or denature the secondary structure of the proteins that are embedded in the cellular membrane of these bacterial pathogens. So at the height of pressure, those proteins are wide open. And then when you release the pressure, it just depends on what else you have in the formulation. It's possible that while those proteins are wide open, if you have acid inside of the formula, or you have other antimicrobial agents, perhaps, they can interact with the hydrophobic core of the protein to the point that when you release the pressure, that protein can no longer spring back into its natural state. So then at the end of the day, what happens is those proteins are sort of denatured. The cell itself can no longer function like it would normally do. And so that's why we usually say in HPP, you get sublethally injured cells immediately after treatment, unless they were already injured to begin with, then those could succumb and go immediately to death. But if you have a lot of healthy cells, they're just sublethally injured. And then we rely on whatever else you might do after HPP. So, for example, freezing could be a stressor to these sublethally injured cells that they continue to die off. Like I said, if you have something in your formulation that continues to stress out these sublethally injured cells, they might just continue to die off as well. So that's very different typically with a heat treatment or thermal treatment. Usually we assume that after thermal treatment, you're completely destroying the pathogen. And so you get a full kill, if you will, as soon as you finish the heat treatment. Let's see. So then that's what's happening on the pathogens. But today we're talking about a virus. So then when it comes to avian influenza virus, think about, well, what is this virus actually made of? It's mostly made of proteins that allow it to bind to a receptor in its host, also proteins that allow it to cleave and replicate. But then inside the virus, inside this kind of protein package, that's where you have the RNA, so some of the genetic material for the virus. So the genetic material can also be somewhat destroyed or affected, potentially maybe just the word disrupted is probably better, disrupted by high pressure. So so far, what we're finding in the literature is most viruses, because of this simpler makeup than pathogens, they're also easier to kill with HPP compared to HPP killing bacteria. So the literature is compared to thermal processing when it comes to HPP. It's a little bit less. HPP, after all, is probably just a 20 to 25-year technology. There's more that we need to do in order to demonstrate its efficacy in a variety of products, a variety of situations, perhaps. The two papers that I've seen and liked about HPP and discussing how it can inactivate the virus, they've done the research with H7N7, which kind of like H5N1 can have a low pathogenic version strain or a highly pathogenic strain. So, some folks might criticize that study and say, oh, it's not H5N1, but I think it probably is going to behave very similarly to H5N1. But the other thing that for that particular study was only demonstrated in ground chicken. So I know some pet food companies have tried to see if they can use that as their validation study. And some of the maybe feedback they've gotten from others was, well, you're not really selling ground chicken or ground chicken slurry. You're selling, let's say, a formulated diet. So that's where we need a little bit more research, again, to kind of demonstrate how well does HPP work in a variety of, let's say, pet food ingredients like chicken livers, maybe beef lungs, because we know cattle can contract the virus and shed large or huge numbers of it, perhaps also in raw milk. I know some raw pet food companies are using raw milk as an ingredient, perhaps. And then we also need to demonstrate perhaps like how sensitive the virus is against different processing conditions. But the good news, like I said, is, the data so far from these limited number of papers show it requires a lower pressure than we would normally use in the US to kill salmonella. Viruses, you need a little bit of lower pressure and then also a much shorter holding time um, to get inactivated by HPP. So promising, but we need the data to really back those up.
Gotcha. So you mentioned a little bit earlier about how formulation can influence
Mm.
HPP. Could you tell us a little bit more about how the composition of a cat food, its fat content, pH, moisture, etc., can influence the efficacy of HPP against the virus?
Yeah, so again, this one is even sparse in terms of the literature when it comes to, if the effectivity of HPP. But we know in bacterial pathogens, there seems to be some kind of a threshold where as you increase the level of fat in a formulated diet, HPP, you can actually help HPP inactivate the salmonella, let's say, in the pet food if you use HPP. However, at a certain point, and we don't know exactly where this certain point is, I think it's kind of somewhere above 15% and below 25%, according to our testing. Um, at some point, the efficacy of HPP starts to drop. And other researchers have also demonstrated this when it comes to RTE meats when they use HPP. When your deli meat has fat greater than 15% and maybe, somewhere before you hit 25%, you start seeing a drop on the efficacy of HPP. So there seems to be some barrier protection or protection from pressure that fat can have. My guess is that probably is going to track similarly when it comes to the virus. So I'm not familiar with formulating pet food diets because I'm not in that world. I'm more on the food safety side rather than on the nutrition and formulation side. But I do think there may be some cat diets out there that have higher than 15% fat content. So for those kinds of diets, we have to start being careful that even if you grab at, let's say, a study done with HPP, for example, the ones that I referred to earlier with ground chicken breast, those will be lower fat. I want to caution pet food manufacturers from extrapolating that data and saying, well, our formulated diets probably are going to do the same. So there will be some requirement of testing to prove or to demonstrate that. pH is interesting because we lower the pH when we start introducing, let's say, some acidulants or acidic ingredients in our pet food diets. So remember me mentioning earlier at the height of pressurization, most proteins will start to unfold and expose its hydrophobic core. So there's going to be positive and negative charges to that hydrophobic core. Your acids, if they dissociate when in solution, they can also then start to attack the hydrophobic core of the proteins that are in the virus. So it is possible then that for in certain situations where you could have the proteins that are in the virus not able to come back down to its natural state at the release of pressure. So they can no longer bind to the receptive host or become infective at all. So they may still be present. The genetic material of the virus might still be present in the pet food, but when it comes to its virulence, it might actually not be working anymore. So I think those are some things that really, I think, great areas for research, some of our knowledge gaps also on how efficacious HPP really could be when it comes to food safety and such. So yeah, there was, let me see. I've read so many papers, I can't remember all of them now. I do know there were some papers out there that tried to see if if the avian influenza virus could be wiped out or inactivated by acids alone. So again, this is maybe more towards sanitation practices, right? Whether or not it's in a slaughterhouse or in a poultry barn. And strangely enough, I think the results were a little bit mixed. You know, it kind of depends on which acid you're using, and then also what's the viral load, because if it really is quite large in numbers, you might not be able to rely on acid alone. But it does shed some light when it comes to your original question for formulation. That yes, pH and acid could work and have an effect on the virus, whether or not we get significant decrease or significant inactivation, that's yet to be seen. So, yeah, there's lots of things that we still don't know.
Gotcha. So just to kind of sum that up, could you just kind of characterize what are the limitations of HPP in activating the virus compared to thermal processing?
Yeah, so because HPP is still a fairly new technology, we need more data to for us to really get a better handle on when is it effective, when it is not. But I think so far the research says that it is promising, that it's going to track pretty well with thermal processing to the point that so long as you have a validated HPP process where you're killing your pathogens, because the virus is a much simpler in structure, mostly made of proteins that encompass an RNA, then you can probably use your validated HPP process right now. If you're able to get a 5 log reduction in salmonella, that it looks like you probably are going to also kill the avian influenza virus. But again, we need more data to really demonstrate that and for us to be able to tick it off right as one of our best practices against the virus.
Gotcha. So kind of a little bit more on that. Like if you were to put together a literature review on
Mm.
research about scientific studies supporting the use of HPP for avian influenza, how much is out there? Is it something that people are working on now?
Yeah, I think that's where some of the challenges are. So if we really want to deal with HPAI, right, like the highly pathogenic strain, we have some limitations. Number one, we do have a couple of universities in the US and worldwide that have HPPs that would be readily available for research. Most of them, however, are in a situation like me, where some of them are in a food grade pilot plant facility because they're using the HPP at the university to encourage more and more entrepreneurs to use the technology. In that particular case, we cannot introduce either low pathogenic avian influenza virus or the highly pathogenic version of the virus to do any kind of testing in a food grade facility because it is, after all, a select agent, especially the highly pathogenic ones. In my case, I do have an HPP, but it is still in a biosafety level 2 laboratory. So we're able to do some testing with low pathogenic strains of H5N1 and then also other LPAI of different subtypes. And that's kind of one of the things that I'm working on is to get started on that. There are other labs. I know of one here in the US, Dr. Alvin Lee at IIT Chicago. His HPP happens to also be connected to a biosafety level 3 laboratory. So then he is able to, if he chooses to kind of validate formulated diets, pet food ingredients, and test and see if certain HPP conditions can inactivate the virus. So right now there's, I guess, we have capacity, but it's somehow pretty low. And then what do you call this? We still, I think there's some struggle still for some of the researchers to have access to the H5N1 virus as well. So even though USDA APHIS has, I guess, provided a reprieve of 3 years to allow more research in this particular area, so they can get the highly pathogenic avian influenza H5N1 virus and request it have it, show up in their lab for testing. But then that version still requires a BSL-3 lab and such. So that's the situation when it comes to research. We were fortunate that we've had some of this papers out there on HPP and HPAI stemming all the way back to the 2015, 2016 outbreak of the virus. But that one was a little bit more short-lived than what we're experiencing now. So kind of the research probably just kind of got dropped, and we addressed other food safety concerns in between.
Gotcha. So for pet food manufacturers who don't have a biosafety level 3 lab that they can
Mhm.
go to in-house, how should those pet food manufacturers validate and verify that HPAI has been inactivated in their products when they're using HPP?
Yeah, so I think for those who are using thermal processes, there's a little bit more guidance, right? They can use WOA, they can use their validated extrusion or canning processes. In this raw pet food space, that I think that's really one of the challenges. So yes, you can probably get a validation study, and then you can probably figure out a way to potentially justify doing it with a low pathogenic strain, or if you really wanted to do it on a highly pathogenic strain. The issue then becomes I think when we get to verification, because we go back to the challenge for testing. We still don't have commercial food testing laboratories able to test for H5N1 on a probably more routine basis where they have to handle large numbers of samples. So back to your question, what else can they do? I think the guidance from FDA Center for Veterinary Medicine, did provide some resources there. I think there were a lot of data when it comes to milk pasteurization, also dealing with eggs. They provided some resources or links to some research out there. One of the things that they also talked a little bit about was supplier controls. And, work with your supplier, figure out if your supplier can attest that they are testing um, for for bird flu, right? Like especially if you're purchasing like a poultry ingredient from them or a beef or cattle based ingredient from them, because that's where testing for the virus right now is really prioritized. So our veterinary diagnostic labs all over the country are working with USDA APHIS to make sure that they can detect any infected flock and prevent it from ever leaving the farm, because that would help mitigate its spread. I know a lot of these veterinary diagnostic labs are also involved in testing for HPAI in raw milk, because they also want to make sure that contaminated raw milk is not freely moving from state to state, so that we're also preventing the spread of the virus through that milk supply chain and stuff. So look further into what your supplier can provide you. And again, I'm mostly referencing that because that was in the guidance from the FDA Center for Veterinary Medicine. They've stated that potentially a supplier control could be one way that a lot of our pet food manufacturers can do or use.
Gotcha. So you'd mentioned a little bit earlier about how HPP can, damage a cell, speaking of bacterias, and then there can be another step that can help completely knock them out. So can HPP be used as a standalone kill step for avian influenza, or should there also be like a multi-hurdle approach there as well?
Yeah, so I think again, once you have the data or if you get a hold of data for your formulated diets, I think what you'll find is yes, HPP alone can probably deliver a substantial kill on the avian influenza virus. It may depend though, because another question that I typically get from people, what it should be looking for. Are we looking for, let's say, a 5 log reduction, or should we be aiming for a higher log reduction on the virus, knowing that the virus tends to replicate very, very quickly and can achieve high numbers very, very quickly? I think there it depends. I know for some of the literature that I saw, numbers on what do you call this? It's a different measure. It's not a colony forming unit because we're not dealing with bacteria. Most of the time, we're looking for something, a high number, 10 to the 4, 10 to the 5. And it's called EID. It's kind of like the measure of the bacteria after the virus isolation and testing in embryonated eggs, chicken eggs. But in any case, if you use that as a measure, I think that the 5 log reduction would be sufficient when it comes to, let's say, if you're using chicken muscle meat or let's say chicken livers and stuff. However, when it comes to milk, raw milk from cows, the number so far that I've heard from someone is the viral loads are so high, it could be 10 to the 10. So now, if you're going to try to use HPP to inactivate the virus in raw milk, all of a sudden you're going to have to challenge HPP to get 10, 11 log reductions. And that's something that, we've never had to do with bacterial pathogens before. So there's another level of challenge. And it's possible, as I mentioned earlier, and I always encourage folks that work with me to kind of find other ways to add to, let's say, your kill steps. So using this multi-hurdle approach, I think is actually really good. Not only do you assure that you have a validated kill step that can deliver the log reduction in that particular food matrix that you're trying to produce and make commercially, but any little thing that you can do along the way that adds a little bit of that kill, it's just kind of like cherry on top on your sundae, right? Like it just gives you a little bit more assurance and such. And it would help you in cases where you encounter a worst case scenario because you don't necessarily always have full control of your product. So if HPP is effective because you assume that immediately afterwards everything is going to be refrigerated, then what if somebody all of a sudden within your distribution fails at doing their job, and then let's say your product succumbs to temperature abuse? Well, as long as you had some of those hurdle technologies, that's where you start relying on them to continue to suppress the growth of the pathogen, or perhaps just continue to suppress or eliminate the virus itself.
And always good to have a backup.
Yeah.
So just a little bit more on HPP, not necessarily about the viruses. How does HPP affect nutritional quality, texture, and shelf life of raw pet foods while ensuring that pathogen inactivation?
Yes. So as I mentioned earlier, HPP is really good at denaturing proteins, at least, changing that conform secondary structure of the proteins and overall its conformation. When it comes to nutritional quality, so for example, some of the vitamins, minerals, amino acids alone, not necessarily the full protein, we're not seeing HPP able to affect those. Sure, there's an effect when it comes to texture, just like cooking denatures your protein. Pressure can also denature protein. In fact, acids can also denature protein, kind of like when you get a steak tartare or a ceviche. Those are not necessarily cooked or pressurized, but the application of acids can render the product safely for consumption if done correctly, but you can also see some discoloration and potentially some texture changes to an animal protein ingredient. So very similar to those other mechanisms of cooking or preparing meats, HPP or high pressures can definitely change the texture. When it comes to shelf life, very similar to how HPP inactivates pathogens. A lot of your spoilage microorganisms, like aerobic plate counts or aerobic bacteria, some of your yeast and molds, especially yeast and molds, are very easy to inactivate with HPP. I think one of the harder ones to inactivate with HPP are your lactic acid bacteria, which over time, if your product tends to spoil, and when it spoils, it will be fermenty, kind of smell of fermentation. The culprit for that might be a lactic acid bacteria. Lactic acid bacteria have a very thick cell wall. They can repair themselves very quickly. They love acidic environment. They love to be refrigerated. So these lactic acid bacteria tend to be harder to kill with HPP. We can certainly reduce their load, reduce their numbers by HPP, so that it takes a much longer period of time for them to grow back up and actually spoil your product. So
Gotcha. So kind of branching off from HPP, are there alternatives to HPP for raw pet foods, and how effective would those be for virus inactivation?
yeah, so I mentioned earlier about acids, right? Because I did look at that, because most of us who deal with, let's say, preparing meats, let's say in a meat slaughterhouse, you see some acid washes here and there. So then I thought, okay, well, let's check and see, for some acids that are typically used in pet food manufacturing, is this going to be a viable option? So far, what I've read is it's mixed. The results are mixed because it really depends. One of the review papers I read said it depends really on which subtype of the virus you're dealing with. Because some subtypes, can actually, because, if they're able to pass through the GI tract, that means they're also able to tolerate the acidic conditions of the stomach, right? Or of the gizzard of the poultry animal. So understanding what those pH values are and whether or not we can actually achieve those acidic conditions, if not worser conditions when we're formulating our pet food diets, we're not sure of. The other one is acids only get you so far. So if the viral load is already very high, then it just might not be enough to stand on its own at inactivating the virus. The good news, like I said earlier, is, we have systems in place where, a lot of our poultry, or not a lot, all of our poultry producers are tasked to make sure they have biosecurity measures so that they're, people working in and out of the facility need to, shower and gown up before they can enter the facility. Also, same likewise when they're exiting the facility, because we're trying to minimize the spread of the virus. Also, we try our best to ensure that no infected flock makes it to the slaughterhouse. So technically, if the meat is already USDA inspected, then we would assume a very, very low risk. It's never zero, because getting to zero risk is really, really hard and almost impossible. But we have a very, very low risk of seeing this virus in meat processing. But I think, again, when you're designing your food safety plan, sometimes you design it for your worst case scenario. So my suggestion there is do not rely on acid alone, because the viral load could be an issue when it comes to its effectivity. And then finally, it's just hard because exposure time, how long do you expose the virus to this, low pH conditions? And is that good for your formula itself? Because you still want to make sure the pet food is palatable. And then let's see, there was another one. I think the type of acid that you use, and we know this for those of you who are working on if you work mostly with antimicrobials, different kinds of weak organic acids have different efficacy against bacteria. So we would expect the same when it comes to viruses. So I talked about acid. There's been some testing on UV treatment as well, because again, when you think about the virus accumulating in the digestive tract, if you really want to test for the virus, feces samples are really going to be high when it comes to viral loads. So any bedding material then in a poultry farm could also help spread the virus all over the flock. So there's been some studies about, well, what if you UV treated some of these bedding materials here and there to kind of just continue to suppress viral loads inside a poultry house. I think the research on that is also a little bit mixed, because some researchers have tried to simulate those conditions, let's say in a biosafety cabinet in a lab. So very different exposure times, different wavelengths of UV can also have different results. But that's definitely something that people have tested. I think more testing in that particular area could be warranted. Because I do know of some raw pet food manufacturers that are using UV to kind of at least pre-treat some of their incoming ingredients. But also just note that UV is a surface treatment. So anything internal, let's say to your 40-pound block of frozen meat ingredient coming in, it won't necessarily pasteurize the whole block. It'll pasteurize the surface of that block unless you decide to apply, let's say, the UV during a grinding process, perhaps. But yeah, so thermal, I think, is still king. We know that those are tried and true temperature time combinations. Pressure, promising. And then some of these other um, technologies that are coming up right now, I think we just need more data on it. And, I guess somewhat sadly, we've never thought of this virus as a foodborne um, kind of virus that we had to deal with, but we are doing it now.
It's certainly on people's minds now.
Yes.
So speaking of what's on people's minds now, I think we've pretty much covered all the questions that we got before the show. Let's see what's coming in now from some folks in the audience. Just to go in a little bit more about the processing controls, are there any that regulatory bodies are suggesting as like a preferred method?
Oh, I don't know about preferred method, but I think all over the board, right, with the World Organization of Animal Health, FAO, CDC, certainly with FDA, CVM, and USDA, FSIS, a lot of them are pointing to the data that we have in the literature in terms of efficacy of thermal processing. Again, that's just because, it's tried and true. We've done, we've always thermally processed our foods. When it comes to non-thermal technologies, we have some few data, but it's just not been demonstrated across a wide range of ingredients and pet foods. But now is the best time for us as I guess as a community to kind of get at those data that's needed, because not only helpful to you, perhaps in the future helpful also to ensure the safety of the human food supply chain, but then it also just kind of adds to our body of knowledge and science, filling in some of these knowledge gaps.
Gotcha. So we mentioned a little bit earlier about there had been an outbreak in South Korea, but most of these have all been in the U.S. and related to U.S. products. Is there any way that pet food manufacturers in the U.S. can ensure international markets of the safety methods that are being used in the U.S. or ensure the safety of products from the U.S.?
Yeah. I think this is where you'll have to work maybe with USDA APHIS a little bit more. I know there have been times in the past, maybe not just pet foods, but also just, poultry meat in general, that if we have a very active outbreak of avian influenza, some countries may reject for a period of time our raw poultry ingredients because they don't necessarily want the virus to enter their countries. For pet foods, again, I think the last I heard, there was a period of time where it was really hard to export, let's say, an HPP treated raw pet food product to other countries because they were wary, it's labeled raw. They don't quite understand HPP. And right now, both the World Organization for Animal Health and FAO don't quite recognize HPP yet as a true way of inactivating avian influenza virus, simply because the data is not yet there, and whatever data that we do have tends to be more on ground chicken slurry or ground chicken breast. And so those are not necessarily what our pet food manufacturers are trying to export. So sorry, that was a really hard question, but there's just not a whole lot of information out there yet.
Not a lot of information about a lot of these things out there. So
I know.
let's see here. I know you also do some work on freeze drying. You
Oh
have a
yeah.
here about mitigating the H5N1 risk related to freeze drying without doing an immediate thorough validation or a traditional kill step. So how does freeze drying relate to HPAI?
Oh boy. Okay, so this is a really good question. So when we actually think of freeze drying, it's really good at preserving, and it's really good at preserving bioactive ingredients. So when you think about, let's say, vaccines or some of the pharmaceutical products, those are actually prepared by freeze drying. So if you're doing a normal freeze drying where, let's say your temperatures inside your freeze dryer never really make it above 120 F. You saw me state the temperatures by WOA earlier. 120 F is not really high enough to inactivate the virus. So freeze drying, think of it more as a preservation technique and not necessarily as a kill step. Having said that, I do know of some pet food manufacturers, or at least I was told, there have been some pet food manufacturers that have tried to get a kill in pathogens inside of their freeze dryers. And so some of them may even be cranking up temperatures inside their freeze dryers all the way to 190 degrees Fahrenheit. So much higher than what we stated earlier, what kind of temperature it takes to kill salmonella and the virus. We were referencing 165 degrees Fahrenheit earlier. So when companies do come to me and say, "Hey, Grace, we're going to get a kill because we're going to go all the way up to 190 F," I usually tell them, "Okay, that's fine, but just recognize your freeze dryer was never really designed to be operated, I would think, at such high temperatures over a long period of time." Number two, because at that point, just use a regular oven. Then the other thing that I would also say is it depends on when you apply the high treatment, uh, high temperature treatment in the freeze dryer. When you do it at the beginning of the process where your product is still pretty wet, your wet product is going to have a high thermal conductivity so that the high temperatures that you're applying on the shelf will pass through the tray and it'll pass through the entirety of your product. So yes, at that particular time, it will definitely kill salmonella. Can definitely kill the virus. However, if you do that at the beginning of the process, it's going to create so much sublimated moisture, pushing it outside of the product. And if your condenser is not able to keep up with all of that moisture being pushed out, you're going to lose vacuum conditions. So then you're going to play this tug and pull with the vacuum pump. So that could also definitely shorten the lifespan of your vacuum pumps and then also your freeze dryer overall. If you do apply 190 F towards the end of your process when your product is dry, then you run into the danger that your thermal conductivity is not high enough. So even though you're heating the shelves at 190 F, your trays are made of metal, so they're still going to reach 190 F, but your pet food product sitting on top is not wet enough to get that 190 F evenly applied throughout the product. So it could be spotty, if you will, when you're trying to use those high temperatures in a freeze dryer at the end of the process, whether or not you're really going to get the kill that you assume or expect to get. So the best thing is, again, as a processing engineer, I want to see equipment and processes used the way they are supposed to be designed. I personally would not recommend using a freeze dryer as a kill step because that's not what they're designed for. They're really, really good at preserving bioactive components. We think of vaccines, those that do have a live virus in them probably also have like freeze drying as a step. It's
Gotcha.
probably way too much of a lecture, but it's one of those things that I get asked a lot.
Right. So when not just using freeze drying as a kill step, but if someone just used HPP or something like that, you mentioned earlier that the viruses can be denatured. Is there an issue with those dead viruses then showing up in like a PCR testing in a laboratory of that virus? And are you aware of any labs in the US that are testing for HPAI in poultry ingredients?
Yes, so let's tackle the first part first. So let's suppose you did denature the proteins in the virus. would that affect your PCR results? So the answer is yes, that's possible. This happens also when we do our validation studies with bacteria. Sometimes, we make sure that we plate and look for viability, that the bacteria is actually still able to grow, let's say after HPP treatment. Because if we relied solely on some of the quicker methods for detection, like PCR, we will still be able to detect little bits of the viral DNA. So this is where it can be very confusing, and you can get a presumptive positive with a PCR test, which kicks it to the gold standard of virus isolation and testing using those embryonated chicken eggs, so that if you were to then grab at the viral samples that you're able to extract and inoculate it into the chicken eggs, that will tell us whether or not did the viral DNA that you were able to detect with PCR, was that virus still alive and can infect chickens, or is it actually truly dead? Now, when it comes to your second part of the question, are there any commercial testing laboratories, whether or not it's for food or, let's say, veterinary diagnostics, that could test for HPAI in pet foods or pet food ingredients? I think right now, almost everybody that I've talked to, and I have looked, I can't find any commercial food testing laboratory that is currently testing for HPAI. There are actually already few commercial food testing laboratories that are testing for viruses overall. Most of them have the capacity to test for pathogens, bacterial pathogens, but not necessarily all viruses. I was told that when it comes to testing pet foods for HPAI, it really depends on the state's Department of Agriculture. So if there is a suspected case of avian influenza infecting, let's say, a cat, a poultry animal, or cattle, then yes, the State Department kicks in. They want to make sure they address that virus being in their state. And then it will be at that time where they can ask the veterinary diagnostic lab in the state to test for the food. But that's what I'm aware of. I'm not aware of any veterinary diagnostic testing labs that are open to testing pet foods from companies that want to send it into them, because that's not what they're designed for. They're designed to look for infected animals or infected flocks. They're also testing milk right now, because all of their work is all about helping to contain the virus and kind of mitigate its spread.
Gotcha. And so here we have a kind of a technical question. If you have a thermal process that is for a lower temperature for a longer period of time, that's been validated for a 5-log reduction in salmonella, then would you also say that that is validated to inactivate HPAI?
Ooh, so that's a really good question. And I've thought about that too. I think right now, just kind of absent of data that is in the literature peer reviewed, I would hesitate saying yes to that one. I mean, I would abide by what WOA says. It looks like, 8.5 minutes at that 120 F was the lowest temperature that they would suggest for killing the avian influenza virus. I think if you go lower than 120 F, then you will probably be required to come up with a validation study that would also demonstrate that. But I would strongly recommend staying within the temperature time parameters provided by WOA at this time.
And a little bit more on that, if whatever procedure if there's a validated white paper for H7N7 bird flu, would that also be valid for H5N1?
Yeah, so I think my understanding, and I'm not a virologist, but my understanding is H5 and H7 tend to behave a little bit closely with each other. The good news right now, though, is, I think, well, two good news is If you're working with a virologist, so let's suppose you are, working with, let's say, UNL. So we do have a virologist on campus, and he told me that it is possible to, let's say, take a highly pathogenic H5N1, and if we wanted to do a validation study, we can convert that or mutate it so that it can lose that one gene that makes it into a low pathogenic H5N1 and use it in our validation studies. So genetically speaking, it almost will look exactly like the highly pathogenic version, but we've removed that gene that allows it to like cleave like crazy and replicate and be classified as a highly pathogenic strain. This is very similar to the strategy used by USDA ARS when they did the beef cooking study last fall. They essentially, they got hamburger meat, 20% fat, made patties out of it, cooked it, either kind of something undercooked, something medium, and something well done. When it comes to burgers, cooked it in an open gas grill, flame grill. And there, they've demonstrated in their study, and this is published now for anybody who wants to get the paper. I can certainly send you the link. That they were able to kill the virus, even though it was actually grilled as a rare hamburger meat and such. So I think there's ways to do the validation study. You want to try to get as close as possible for H5N1, but I do believe there's enough data out there that also says that, let's say, an H7N7 low pathogenic one would behave very similarly to H5N1.
Gotcha.
Yeah, I do want to add to that. Remember for your validation studies when you're doing bacteria, you typically would have a cocktail of different strains of the bacteria because you want to make sure you encompass within a particular space as many of the different types of salmonella that your product could potentially encounter in the future. And so you want to demonstrate that your process is robust against this wide range of bacteria. So I think what would help the industry overall is if when we're able to do validation studies with avian influenza virus, that we also go towards the same similar approach where we test the efficacy of, let's say, HPP, let's say acids, let's say potentially some of the sanitizers that we're not sure if we should be using them. When we test for efficacy to control this virus, I think we should also not hyper focus on just the highly pathogenic H5N1 only, but we should use the approach of trying to understand how it could work for a larger range of avian influenza virus subtypes.
Gotcha. All those viruses. So you mentioned there acids and using that. We have a question here about would you recommend organic acids or a combination of organic and inorganic acids? And are there any specific acids that are proven effective against avian influenza?
Yes. So, like I said, the literature on this is a little bit mixed. I don't remember exactly which ones from my reading were most effective or not effective. And then all those other factors that I mentioned earlier, which particular avian influenza subtype, viral load, which type of acid, and how long is the exposure time? All of these factors have a confounding effect, right? So you have to almost treat this whole matrix and come up with a multi-level treatment experiment, if you will, to kind of truly understand which acids are going to be effective or not.
All right. So speaking of ingredients that might be at risk, is poultry meal at any risk of carrying avian influenza?
Yes, so my understanding is poultry meal is a byproduct of the rendering process. So in this particular case, the high pressures and high temperatures used during rendering should be very similar to, we should see effectiveness very similar to what we saw with extrusion and retorting, where both of those are also using a combination of high pressure and high temperatures. So long as, again, the rendering process has been validated to inactivate salmonella and your other bacterial pathogens, then I do think it's also inactivating the avian influenza virus. So I think poultry meal is safe, but we always have to worry about potential cross-contamination that could occur during manufacturing, right? So even though you may have a safe poultry meal, what other ingredients are you using, and what are some of the other things you're doing in the plant to prevent potential cross-contamination.
So following up on that idea of cross-contamination, has there been any research done on possible pathways for reintroduction of HPAI through the environment post-processing, via employees, equipment, cross-contamination, or poor sanitation?
Yes, so I'm not sure of any formal studies yet. I haven't really kind of really looked into that. But just again, looking back at what are some of the measures that we're doing in the poultry houses, right? The fact that their workers have to, make sure that they don't have contact with wild birds. Again, all the preventive measures that they're doing to prevent the spread of the virus. I think some of those we can also think and look into whether or not that's practical and possible for us to implement when it comes to processing. I've now seen some guidance documents too, like at home when you are preparing food for your companion animal, to make sure that you wash your hands and that you're kind of doing it in a sanitary way, so that you yourself as the food preparer are potentially not introducing the virus into their food. So, because even though you might have strictly indoor cat, you yourself are hopefully not strictly indoors. So it could be possible that you're bringing all sorts of other viruses, or, just bacteria in your hands, because you have a very active lifestyle, back into the home. So you want to make sure your hands are clean.
Don't go handling any wild birds.
Yes, right.
So, one of our listeners is curious about the effectiveness of medium chain fatty acids on avian influenza or other pathogens, including salmonella.
Okay, so this is really not my area. So I don't have a whole lot. I don't have any like guidance on that one. So I apologize.
Alrighty, and it looks like we are now down to our final question in the list here. Is Oh, okay, that wasn't even a question. That was just a comment. All right, good deal. So, those were some excellent questions, and we have gone over time here. So I thank you for spending a little extra time with us. And did you have any final thoughts, Grace?
Yeah, so I think overall, I'm really happy to see this many people in the audience. It definitely is on top of our minds. My suggestion is to, as a group, especially for the raw pet food manufacturers, to try to collaborate a little bit better, right? Because we're all in this. Food safety should not be a competitive advantage at this particular point in time. We ourselves in the pet food space want to make sure that we're not spreading the virus, so that we as a country can kind of get a hold and control of this particular virus. I'm happy to share any of the references that I kind of referred to this talk. So just email me. You can look me up by Googling my name and also going to the UNL website. Thank you, Tim. This has been a fun discussion. That was a little bit scary, but a little fun discussion.
I think avian influenza is a little bit scary, so
Yes.
that's fitting. All right, thank you, Grace, and thank you to our audience for joining us. We would greatly appreciate your feedback on today's chat. So please scan the QR code that you should now be able to see to access that survey. We'll also email a survey link very soon. And I'd like to give a great big thanks to Extra for sponsoring this edition of Ask the Pet Food Pro. Please watch for more Ask the Pet Food Pro chats brought to you by Pet Food Industry and Pet Food Forum. I'm Tim Wall, Senior Reporter for Pet Food Industry, and I'd like to say thanks again to Extra and Dr. Mary Grace Denow, Research Associate Professor in the Department of Food Science and Technology at the University of Nebraska, Lincoln. And also, a great big thanks to the Watt Global Media staff for making this possible. Thank you to our international audience for joining us. I hope you all enjoy the rest of your day or evening, depending on which hemisphere you call home. Until the next time on Ask the Pet Food Pro, goodbye.